So, good morning to all of you students of BT First Sem. Uh, as I have mentioned in previous day that I will discuss uh, about the gene therapy. I have already started the lecture of the ex vivo gene therapy, and we were continuing with the vectors that are used in ex vivo gene therapy. So let us try to finish this ex vivo gene therapy in today's lecture. So we have done up to the retrovirus as a vector for delivering the genes for gene therapy. And now today I will discuss about the human artificial chromosomes and the what can I say bone marrow to be used in the gene therapy. So let's get started now. So here it has been mentioned about the human artificial chromosomes. What does it says here? Let us see. It is saying here that human artificial chromosomes, in short, we say them as HAC, is a microchromosome that can act as a new chromosome in a population of human cells. So, what does human artificial chromosome is? What does human artificial chromosome is that it is a microchromosome, a small chromosome, okay, that can act as a new chromosome in the population of human cells. So what does microchromosome means actually? Microchromosome is a type of very small chromosome which is typical component of the karyotype of birds, some reptiles, fish and amphibians. Generally, microchromosome is also present within the uh, genome, but we are preparing a microchromosome, an extra microchromosome, and within that microchromosome we are trying to insert our gene of interest for the purpose of gene therapy to cure a certain disease, either by inactivating certain genes that are causing the disease, by knocking out, that means we can say, or by to insert such genes into that chromosome to be introduced into the target human body so that that chromosome may function, okay, as a normal chromosome, and the gene will also do continue to do their function normally, okay. And generally microchromosomes are less than 20 MB in size. It is less than 20 megabase pair in size. But here the microchromosome that we are preparing, okay, the human artificial chromosome that we are saying that it is a microchromosome, it is up to 6 to 10 MB. Okay, so so up to 6 to 10 MB or megabases in size, these human artificial chromosomes are prepared. Okay, so instead of 46 chromosomes in human, the cell could have 47 chromosome. The 47th chromosome is a microchromosome, which is your human artificial chromosomes. Generally, natural chromosomes are of many large base pairs, about 50 to 200 MB base pairs contain. But here, the microchromosomes that we are preparing for human artificial chromosomes is roughly up to 10 megabase pairs. Okay, and these human artificial chromosomes will carry new genes introduced by the human researchers. Ideally, researchers could integrate different genes that perform a variety of function, including disease defense. So, suppose a certain genetic disorder occurs in particular human beings. Now, by using this human artificial chromosome, we can able to introduce them, and we can able to express that normal gene so that that dysfunction can be eliminated, can be removed out. Further, we have previously we have said about a gene augmentation therapy, gene inhibition therapy. So we are also applying that same technique by using this artificial chromosome as a vector. So here we are using this artificial chromosome as a vector, as a carrier molecule. Okay. But what is the main uh, attractive feature? The main attractive feature for creating this human artificial chromosome is that this human artificial chromosome can be passed down from generation to generation when inserted during in vitro fertilization stage. So that's a great advantageous feature for human artificial chromosomes, that the chromosomes can be can be introduced at the in vitro fertilization stage. Well, I think you have studied about the in vitro fertilization, where we use a surrogate mother for uh, conceiving that matured egg, matured zygote, which is normally manipulated within the laboratory. Okay, at that time we can able to introduce this microchromosome, and that chromosome containing desirable functional genes can be expressed into the next generation. But that is where that will become an ethical issue. Nowadays we are using this human artificial chromosome for the somatic cells only, so it is confined to that only on that generation, not it will pass on to the next generation. However, if we introduce this human artificial chromosome in germ cells, okay, that is in the sex cells, then only can able to pass on. Okay, now to prepare human artificial chromosomes, there are two models. So there are two accepted models which can be used for creation of human artificial chromosome as a vector. Now, how we normally prepare these human artificial chromosomes? We have to say this. The first method is the first method is to create a small mini chromosome by just altering a natural human chromosome. So we will take out a normal human chromosomes, okay, from certain cell, and then we will alter that chromosome by removing out non-essential part of that chromosomes, and in that place we will insert our gene of interest for the purpose of gene therapy, and then we will introduce them into the host cells, because the natural chromosomes will have all the essential regions. It will contain the telomere sequence, it will contain the centromere sequence, right? It will also contain the repetitive sequence that we have studied in VNTR, variable number of tandem repeats, short tandem repeats that are normally found in the chromosomes. That's why it is not too much hard to prepare human artificial chromosome by this method. So we are just actually isolating a small mini chromosomes. We are first creating, we are actually isolating a normal human chromosome, and then we are removing out the non-essential part from it, and then we introduce our desirable gene or genes into that chromosome. Okay, and it can be only be accomplished, that means it can be done only by truncating is a good word. Truncating means we are actually cleaving, we are cutting, we are removing out the some of the segments of the natural chromosome, followed by introduction of unique genetic material via the Kree-Lock system of recombination. So by the recombination technique, first what we do, we first isolate the normal chromosome, okay, from a particular cell, then we truncate that natural chromosome, we remove out the unessential segments from that chromosomes. Okay, here unessential segments means those which are not required for that chromosome stability. Okay, okay, and in that place we will now introduce our genetic material. Okay, and how we will introduce our genetic material? With the help of Kree-Lock's system of recombination. Well, Kree-Locks is a type of site-specific recombination. Um, so it is saying here, I have already discussed with, I have just little bit discussion I have done in these notes. That Kree-Locks recombination is a site-specific recombination used to carry out deletions, insertions, translocations, and inversions at specific sites in the DNA of cells. So it is a site-specific recombination technology. With the help of Kree-lock system, we can able to remove out certain segments from here from the chromosome and insert our unique genetic fragments which are meant for curing certain disease through gene therapy. Okay, now site specific recombination is done by through recombinase enzyme. So site specific recombinase technologies are the genome editing tools that are depend on recombinase enzyme. So these recombinase enzyme are specifically used to replace the target sections in the of the DNA. Okay, so simple thing. The first method is very simple. We have to isolate our natural normal chromosome from a particular cell, not from the host. Okay, and we are creating the human artificial chromosome from a natural normal cell. We are then removing out the unessential segments from that chromosome. Here are the unessential segments are those segments which are not required for their step for the stability of the chromosome, like we can remove out the repetitive units, we can remove out the all the other genes. Okay, and in that place we will introduce our desirable gene via recombination technology, via site specific recombination. So if you're not able to write down Kree-Lox recombination, you can write site specific recombination, SSR. So through site-specific recombination, we can introduce our gene in within that chromosome, and then we will target that chromosome into the specific cell of that host. Okay, so this is the first method that generally we use for preparing the human artificial chromosomes. The second method is we are creating the artificial chromosomes actually. So the second method involves literal creation of a novel chromosome, a de novo. De novo means from the beginning, that means new. So we are creating a new artificial chromosomes. Okay, this is a second method. So de novo we are normally using for the creation of a novel chromosome. But for the creation of a new chromosome within the laboratory is a difficult task, because the we need centromere region, we need telomere region. So these are some essential region where we will find this centromere region, where we will find this telomere region. Obviously from the normal chromosome, not telomeres, telomere regions. Okay, so these are required for making an artificial chromosome. So this is the second method. Now artificial chromosomes are highly promising vectors. So here we are using, instead of using virus as a vector, as we have discussed previously, retrovirus as a vector, here artificial chromosomes can be employed as a vector, as a carrier molecule for the use in gene therapy applications. They are able to maintain expression of genome-sized exogenous transgene within the target cell. So here we can able to, with the help of artificial chromosomes, we can able to express our desirable genes. Here exogenous transgenes means, exogenous means from outside, transgenes are those genes which are from outside, we are incorporating, we are trying to express that genes into the cells via this vector, via this human artificial chromosome as a vector, without integrating into the host chromosome. So this is a second advantage, because here we are not altering the human genome, we are not altering the human genome, we are sorry for that, we are altering the human genome, but we are not integrating the gene into the human genome. We are just making an extra chromosome, okay, artificially, then we are inserting them into the genome. We are inserting them into the nucleus of the target cells. Okay, So there that chromosome will be expressed separately. So here we are not trying to modify the genome. Okay, here we are not trying to insert that gene into the genome so that there will be a chance of loss of nucleotides, right? Essential nucleotides, that where it will bind. But we are actually directly, we are separately, we are expressing that mini chromosome within that target cell. Although these vectors have huge potential benefits when compared against normal expression constructs, they are highly complex. Obviously, to make a artificial chromosome is quite complicated, technically challenging to construct and difficult to deliver to the target cell. The main problem arises when we are using this human artificial chromosome is first when we are constructing, that how we will construct. Okay, we have to maintain that structure stability of the chromosomes, otherwise when we will target them into the cells they can get degraded. So for their stability we need proper telomere sequence, proper centromere sequence, which are essential for stability of a chromosome. And at the other hand, we have to properly deliver that chromosome into the target cells. Okay, now big chromosomes are difficult to deliver. Small chromosomes are easy to deliver. But suppose we are using such gene segments which are quite large in size, then it will be difficult to transform. It will be difficult to transfer into the target cells. So that's some problems will arise while we are using a human artificial chromosome as a vector. Now I have taken a diagram from an internet source that you can see here. Okay, so this above figure is the normal chromosome, while the figure that you can see, small figure in below, is the human artificial chromosomes. Here you can see that the telomere sequence has been taken from a normal chromosome and constructed to form the telomeres of the small mini chromosome. Okay, then the centromere region is required because these are the unique sequence na, and it is to be introduced into the hair for the human artificial chromosome. And the blue ones that you can see here, these blue ones are your essential targeted gene, okay, which has to be what can I say, that has to be used as a that has to be used for the gene therapy actually. Now this small chromosome will now be targeted to the target cells where this chromosome will be expressed. They will perform they will, just like mitosis, meiosis we have seen, they will perform the same thing. Okay, so this is a good example explanation of how a human artificial chromosome is actually made actually. Okay, so I hope you have understood about this human artificial chromosome. Let us move on to the next page. Okay, now comes bone marrow cells for gene therapy. Bone marrow cells for ex vivo gene therapy. What it is saying, let us see now. Bone marrow consists of a totipotent embryonic stem cells. So within the bone marrow, okay, um, within the bone marrow that is found inside a long bone, these bone marrows actually are having a totipotent embryonic stem cells. Well, totipotent means having the ability to create the different types of cells, just like we have seen in case of when we are talking about the agricultural biotechnological applications. I have said little bit, mentioned about the totipotency of plant cells, that plant cells have the ability to give rise to any cell type. That's why they are totipotent. But unlike plant cells, animal cells, all the cells are not totipotent. Here, the embryonic stem cells are totipotent. Well, embryonic stem cells contain within the during the fetal stage, like the blastocyst stage. There we get embryonic stem cell from the inner cell mass, and here also from the bone marrow also they are also contain embryonic stem cells which have a totipotency in nature. That means they are capable of dividing and differentiating into various cell types, right? So any type of cell can be prepared from this embryonic stem cell, like red blood cell, like blood platelets, like macrophages, like osteoclasts, like B lymphocytes and T lymphocytes, which are involved in cell mediated immunity. And for this reason, bone marrow transplantation is most widely used technique for genetic disease. This is the main reason that why we transplant bone marrow, okay, for curing certain genetic disease. Now this required a lot of money. Obviously cost factor is an important limitation for bone marrow cell in gene therapy, because we have to transplant a new bone marrow, um, uh, a short fragment of bone marrow having that normal gene to correct that human disorder. And why we are using bone marrow, that I have said, that bone marrow content totipotent embryonic stem cells that can give rise to any cell types like red blood cells, platelets, macrophage, B lymphocytes, T lymphocytes. That's why bone marrow transplantation is being done nowadays for for curing genetic disease. And there is every reason to believe that genetic disorders that respond to bone marrow transplantation are likely to respond ex vivo gene therapy. Now we normally go for ex vivo gene therapy when we are using the bone marrow transplantation actually, like from the table 13.2 it has been given, where with the help of bone marrow, we have we have used this ex vivo gene therapy for curing certain genetic disease. Let us see that in the table. Yes, this is the table. So, selected list of genetic disease that are likely to be cured using bone marrow cells. Okay, like this disease I am going to discuss with you now. Severe combined immune deficiency, called as SCID. This is a serious disease which will disrupt your immune system. Well, to cure that disease, we need a normal gene, okay, that are causing this disease due to due to lacking the function of a certain gene. The name of that gene is ADA gene, okay, ADA gene. This ADA gene make a protein actually called adenosine deaminase. Adenosine deaminase. This gene gets non-functional for the people who are suffering with SCID. So we have to use a normal ADA gene. We have to introduce a normal ADA gene via ex vivo gene therapy through bone marrow for curing SCID. Okay. Then another example is sickle cell anemia, Fanconi anemia, thalassemia, most of the blood related problems, Gaucher disease, Hunter's disease, okay, Hurler's syndrome, chronic granulomatous disease, infantile agranulocytosis, osteoporosis. These are common genetic disorder that is nowadays occurring in the Western countries. X-linked agamaglobulinemia, X-linked agamaglobulinemia. Okay, this is another type of genetic disease which occurs on the sex cells actually. Okay, so all of these genetic disease can be cured through bone marrow uh, by the ex vivo gene therapy. Okay. So there is an example given, for instance, that if there is a gene mutation that interferes with the function of red blood cells, which causes sickle cell anemia, bone marrow transplantation is done. For example, has been given that sickle cell anemia occurs why? Because due to mutation within the gene of red blood cells that leads to sickle cell anemia. And how to cure that sickle cell anemia? By introducing a bone marrow that will contain that correct gene, that normal gene, non-mutated gene. Okay. Bone marrow cells are the potential candidates for gene therapy for sickle cell anemia. However, this is not as simple as theoretically stated. Well, theoretically we are discussing here. It is like it's quite simple, right? But it's not so much simple if we do practically actually while doing these transplantation techniques, because uh, many parameters has to be seen here. While we are transplanting the bone marrow, we have to immune suppress them so that this bone marrow will not likely to get uh, what can I say, a term content na, a term say we say na, in case of skin transplantation we say na graft rejection, that type of thing can occur. Okay. Our own the host immune cells will identify that transplanted bone marrow as a what, as a foreign cell, so it will as antigen, it will kill them, it will destroy them, just like it occurs na in case of heart transplants or skin transplants. Okay, where the patient has to be kept in immune suppressed condition by using immune suppression drugs. Okay, so that type of parameters, I have said only one thing, more many other factors has to be regulated, has to be taken in care while doing the bone marrow uh, gene therapy. So it's not easy as we are seeing here. Okay, that using the bone marrow we will isolate the bone marrow from a normal cell, from a normal organ of a certain tissue of a target cell, and then we will isolate the cells that contain that desirable gene, and we will introduce that cells into the patient's host's body in bone marrow, where they will try to perform them normally. But that this is not as simple as it is. Okay. Okay, now I have to mention a disease which can be cured through ex vivo gene therapy. The most common that studied among the students in your graduation is your severe combined immune deficiency. So this disease is normally a immune deficiency disorder. It will disrupt your immune system actually, and it occurs due to deficiency of due to mutation of a certain gene that responds to your immune system, which is your ADA gene. So what it is saying, let us discuss now. It is saying that severe combined immune deficiency, or SCID, it is saying here that this is a rare inherited immune disorder associated with the T lymphocytes, and to a lesser extent B lymphocytes dysfunction. So what does SCID means? SCID, what does SCID occur? Refers to dysfunction or non-function of T lymphocytes and to some extent B lymphocytes. Well, we know that T lymphocytes and B lymphocytes are involved in cell-mediated immunity, right? About 50% of SCID patients have a defect in the gene located on chromosome number 20 that encodes for adenosine deaminase. So it is saying here that what can I say? It is saying that those patients who are suffering from SCID, they have a mutation in their gene that is located on chromosome number 20. Right? And what is that gene which is located on chromosome number 20? The gene which gets defected, the gene which gets defected in chromosome number 20 is your gene which will code for the adenosine deaminase. So the gene which is responsible for adenosine deaminase, if it gets non-functional, then it will leads to SCID, that causes dysfunction or non-function of T lymphocytes and B lymphocytes. Okay, now in the deficiency of ADA, deoxyadenosine and its metabolites, primarily deoxyadenosine 5' triphosphate, accumulate and destroy the T lymphocytes. Let us try to figure out what they are trying to say. I have simply said that adenosine deaminase is a enzyme, right? Now this enzyme is coded by certain gene which is located on chromosome number 20. Okay, which is located on chromosome number 20, and due to mutation of that gene, that is responsible for adenosine deaminase, it leads to the formation of that SCID disease. Okay, now what does adenosine deaminase do? When adenosine deaminase get non-functional, deoxyadenosine and its metabolites like your deoxyadenosine 5' triphosphate, they get accumulate and destroy the T lymphocytes. Normally, deoxyadenosine 5' triphosphate will be broken down by adenosine deaminase enzyme. So, this deoxyadenosine 5' triphosphate will not get accumulated, will not get collected within the T lymphocytes, because adenosine deaminase is at that time is functional. But suppose if this adenosine deaminase enzyme become non-functional due to mutation, due to change in the nucleotide bases, then what will happen? This deoxyadenosine 5' triphosphate and uh, other metabolites related with deoxyadenosine will get accumulated, will get collected within the T lymphocytes and will destroy the T lymphocytes. Well, we know that T lymphocytes are very essential for the body's immunity. Besides participating directly in the body defense, these T lymphocytes promote function to the B lymphocytes to produce antibodies. Well, you have studied uh, the immune system in class 12, that T lymphocytes also are responsible for the activation of B lymphocytes to generate antibodies, well, during cell mediated immunity. Now, if T lymphocytes get damaged due to accumulation of deoxyadenosine and their metabolites like your deoxyadenosine 5' triphosphate, they gets accumulated, they gets collected within the our lymph system, within our blood, and it destroy the T lymphocytes, and thus T lymphocytes will not activate the B lymphocytes. In this way, our cell mediated immunity becomes stopped. Okay, so to rectify this disease, to correct this disease, to cure this disease, we have to use a normal ADA gene, because due to the deficiency of the ADA gene, that codes for adenosine deaminase, these things are happening. So the patients of SCID lacking the ADA gene suffer from infectious disease and die from at young age, because if the immune system get disrupted, then you can be killed by normal cough and cold. Okay, so just like your HIV, in HIV what happens, in AIDS what happens, they disrupt the T lymphocytes, especially the Th cells, the helper T cells, right? Here in HCID what happens, here no infection is occurring here due to mutation of the ADA gene, that codes for adenosine deaminase enzyme. It leads to the accumulation of the deoxyadenosine and their metabolites, like your deoxyadenosine 5' triphosphate, to be accumulated within the blood and destroy the T lymphocytes. And in that term, T lymphocytes cannot able to activate the B lymphocytes. In this way, your body immune system gets totally, say the cell mediated immunity gets totally blocked, and the patients will die at young age by other opportunistic infections. Previously, the children suffering from SCID were treated with conjugated bovine ADA or by bone marrow transplantation. So, previously, the patients of SCID who are lacking the ADA gene, they suffer from infectious disease and die at young age. And in previously, the children who are suffering from SCID were treated with a conjugated bovine ADA gene. That means the ADA gene, which is isolated from the cows, from the bovine, were actually injected into the patients who are suffering from SCID, or we can use bone marrow transplantation for curing that SCID disease. Okay, now let us see the technique for gene therapy for curing the SCID disease, for curing the SCID disorder, better to say. So, the general scheme of gene therapy adopted for introducing a defective gene in the patient has been depicted. Okay, the same procedure is suitable for modifications, can also be applied for other gene therapies as well. Well, by using this diagram, when we will see, the same procedure is applied for curing the other disease. Okay, let's see that diagram. So here, this is the whole diagram. I am smalling, then I will zoom it into one by one. This is the whole diagram. You can take a screenshot to see them. So this is the patient having SCID, which is suffering from SCID. That means its ADA gene is disrupted. That's why the patient is not not able to produce the T lymphocytes, and their immune system is totally blocked. So what we do actually, we isolate we isolate specific tissues. We isolate specific tissues from that patient. So we are isolating the specific tissues uh, from the patient who is suffering from the SCID. Here we isolate the lymphocytes, okay, from the patient who is suffering from SCID. Then what we use is we use certain vectors here. They use virus as a vector, retrovirus as a vector, which contain your, you can see here, if I zoom it in, you can see here within the vector DNA we have introduced the normal ADA gene, okay, incorporated within the vector, virus vector. And thus this retrovirus having your desirable ADA gene. Now this virus is going to be transfect. That means it is going to infect with that lymphocytes. So these vectors is going to transfect or infect with the lymphocytes with these vectors. And we will culture them within the laboratory to check that which of these cells have our desirable vector inserted. That means if a vector is inserted, that means your ADA gene has also been incorporated and will get expressed within the target cells. So growth of cells is done. So here you can see culture cells to verify the expression of ADA plus transgene. So the ADA gene is going to be expressed now. Okay, okay, there's a possibility now these transfected cells is going to be introduced again into the patient, within the target tissue, where from where we have isolated that lymphocytes target cells. The same lymphocyte that now the lymphocytes have become modified, we have introduced our desirable gene via vector. It is again reintroduced into the patient and we express them, and thus the patient after certain days we check for their that whether they are producing normal ADA gene or not. So then they will see that they try to synthesize the normal ADA protein, where we will rectify the disease, we will correct the disease, will cure the disease. So what it is saying here, let us see the same thing has been given. So it is saying that a plasmid vector bearing a proviral DNA is selected. So first, what we do, we use a plasmid vector that have a proviral DNA. So a proviral DNA is being used. The part of the proviral DNA is replaced by the ADA gene. So a section of the proviral DNA is omitted out, and in that place we introduce our desirable ADA gene here, which will encode for the human adenosine deaminase enzyme. And we have also used a selection agent also, like a gene G418, which will code for ganciclovir, which is the antibiotic resistance gene. And then the whole thing is cloned. Okay, now this antibiotic resistance gene will help to select that which of the cells having our vector properly inserted. So again I am repeating, here what we have seen, here we have seen that the vector DNA, we have isolated the cells from the patient. Here the lymphocytes has been isolated. Then what we use, we introduce this vector into that target cell. Now before introducing, we actually use, here the vector is used as proviral DNA. Here we use a retrovirus. Now within that viral DNA we have removed out certain non-essential segments which are not required for the viral replication, but we have introduced and we have in that place we have introduced the ADA gene, the human ADA gene, and also we have also used a certain selectable marker gene for proper selection, that when after we have introduced this viral vector into the target cells, we have to check na that which of the cells are having that desirable DNA or not, desirable vector or not. For that we use a selective marker gene also, like here we use G418, which will provide you antibiotic resistance. Okay, when you will grow these target cells into a antibiotic medium, certain antibiotic medium. So here we can see, and then we will express them. Okay, now this antibiotic resistance gene will help to select the desirable clones within the ADA gene. So circulating lymphocytes are removed from a patient suffering from ADA deficiency. Now these lymphocytes are then removed. So what we do, what we have done, the lymphocytes which the patients who are suffering from SCID, we have isolate their lymphocytes as given in the diagram. Then these cells are transfected with ADA gene by exposing to billions of retrovirus carrying the said gene. So above mentioned gene, the lymphocytes are now transfected with the vector DNA. Here we use retrovirus as a vector containing our desirable ADA gene that is mentioned in the diagram. The genetically modified lymphocytes are then grown in the culture medium to confirm the expression of ADA gene and then return to the patient. So then we will culture them, we select them, and we check them that whether the ADA gene is getting to be expressed or not within the culture cells. If we see that appropriate concentration of ADA gene is going to be expressed within the culture cell, then we are going to reintroduce them into the patient. These lymphocytes persist in children and persist in circulation and synthesize the ADA, and started to synthesize the ADA within the patient after introducing. Consequently, the ability of the patient to produce antibody is increased. However, there is a limitation. So obviously, there's a limitation, and the limitation have a short lifespan. Lymphocytes have a short lifespan, just lives for a few months, and hence transfusion has to be carried out frequently. So the major limitation of this approach is that the lifespan of lymphocytes have a very short lifespans, and therefore it has to be, we have to repeatedly we have to transfuse them. Okay, so transfusion is has to be done oftenly. Okay, frequently we have to do for curing this SCID disease. Okay, now another approach is there for curing SCID disorder is if we can able to transfer this ADA gene into the stem cells. What will happen? Transfer of ADA gene into the stem cells. Stem cells are those cells which are pluripotent in nature, which can give rise to any cell types. So what happened in 1995? ADA gene was transferred into the stem cells, and the stem cells were obtained from the umbilical cord blood at the time of baby delivery. So when during parturition, okay, during the birth of a baby, the umbilical cord which joins the mother's womb with the baby, that umbilical cord contains stem cells, as we know. That stem cells were isolated, and within that stem cells we transfer that ADA gene into that stem cells via retrovirus as a vector or via human artificial chromosome. Anyhow, through the help of carrier molecule vector, we are introducing the ADA gene into the stem cells. 4 days after birth, the infant received the modified cell back. By this way, permanent population of ADA gene producing cells can be established. So suppose SCID patient who are suffering, they can transfer them to the young ones. What we have done, we have isolated the stem cells from the umbilical cord, and then we modified the stem cells by introducing the ADA gene via vector, and then we again reintroduce them into the umbilical cord at the time of baby's delivery. 4 days after birth, the infant received modified cells back, and in this way, permanent population of ADA gene producing cells can be established. Okay, so in this way, we can treat them. Okay, there are also other therapies which can be involved, like this one. Okay, let us see this therapy. Lesch-Nyhan syndrome. Lesch-Nyhan syndrome can also be cured by ex vivo gene therapy. Lesch-Nyhan syndrome is an inborn error in purine metabolism due to the defect in the gene that encodes for the enzyme HGPRT. The full form of HGPRT is hypoxanthine guanine phosphoribosyl transferase. So Lesch-Nyhan syndrome is occurring due to error in purine metabolism. Your purine formation does not occur. The purine metabolism pathway is defected due to the defectness in the gene. The name of the gene is HGPRT, and that gene will encode the enzyme called hypoxanthine guanine phosphoribosyl transferase. If there is absence of this enzyme, then purine metabolism will be disturbed and uric acid level will build up, resulting in severe gout and kidney damage. So as purine metabolism is inhibited due to the absence of this enzyme HGPRT, then what will happen? There will be accumulation of the uric acid, and that result in severe gout and kidney damage. Those victims of Lesch-Nyhan syndrome exhibit symptoms of mental retardation beside urge to bite lips and fingers and cause self multitation. Okay. So Lesch-Nyhan syndrome can also be cured by gene therapy. And why Lesch-Nyhan syndrome occurs? Due to defectness in the purine metabolism, due to non-functional of HGPRT enzyme, hypoxanthine guanine phosphoribosyl transferase. This HGPRT enzyme is involved in purine metabolism. Now to correct this to rectify this disorder, what we have to do is we have to use a normal HGPRT gene which will code for this hypoxanthine guanine phosphoribosyl transferase, that can be introduced via vector and introduced to the target cell of the patients, culture them within the laboratory, and then again reintroduce that cells into the patients again, just like we have done in SCID. Okay. Then other things are also there. Okay. There's another one called familial hypercholesterolemia. Hypercholesterolemia. What does it occur? Why does it occur? Patients of hypercholesterolemia lack the low density lipoprotein receptors. So within their liver cells, there should be a low density lipoprotein receptors which are used to metabolize the low density lipoprotein, which is called as LDL. Now, if low density lipoprotein receptors are not present on the liver cells, that means the gene responsible for that receptor formation is somehow non functional, becomes inactivated, and does this low density lipoprotein cholesterol does not get metabolized in the liver, and it gets accumulated within the blood circulation, and that can cause heart blockage, that can cause heart different type of artery blockage and heart disease. So hypercholesterolemia is a complicated disorder where we can see that due to the lacking of the receptor called LDL receptor, low density lipoprotein receptor, in the liver cells is lacking. It is not present, and due to which this LDL cholesterol is not able to metabolized by the liver. It gets accumulated within the blood and cause blockage in the arteries, blockage in the heart, right? So the gene responsible for that receptor has to be introduced via ex vivo gene therapy. So attempts are being made by gene therapists to help the victims of familial hypercholesterolemia. In fact, there is some success also. In a woman, 15% of the liver was removed. The hepatocysts were transduced with retrovirus carrying the gene for LDL receptors. These genetically modified hepatocytes were again infused into the patient liver. So simple thing, uh, like in previous days, what they have done, 15% of the liver from the patient was removed who is suffering from the hypercholesterolemia, and then these hepatocytes which are the liver cells were transduced, means introduced by, with the help of retrovirus we introduced the gene responsible for that LDL receptor formation, and we check them that which of these hepatocytes are transfected, have properly expressing that LDL receptor gene. Then these genetically modified hepatocytes were again reintroduced into the patient's liver again. So very easy thing. It seems like, but it's not so much simple if you go for practically. Okay, so the gene responsible for the LDL receptor is to be introduced via a vector into the patient's hepatocytes, which is your liver cells, and that can be done by isolating the liver cells from the patient. About 15%, 15%, 15% of the patient's liver cell is isolated, and there we have tried to transfect that liver cells. We have introduced the gene responsible for the LDL receptor into the cells of hepatocytes from the patient's liver that was isolated, and then we check them for its expression, that whether the gene is getting to be expressed or not within the laboratory, and then in vitro, and then we will introduce that modified hepatocytes into the patient's liver again. Okay, now it is saying the hepatocytes establish themselves in the liver and produce functional LDL receptor. So this is such so much of success that we have seen. Okay, okay. Now nowadays, certain improvement is done in the patient's condition. Okay, by observing the lipid parameters in the blood was observed, so that no such type of rejection should not be done. Okay, so these were all about your ex vivo gene therapy in treating various disease. There is also another thing, hemophilia. Okay, we can also treat hemophilia patients by this ex vivo gene therapy. Well, hemophilia occurs due to the non-function of this protein called clotting factor 9. So the gene that is responsible for the clotting factor 9 is non-functional, and that is leads to excessive bleeding. No clotting will occur, and the patient will die due to loss of blood. Now through gene therapy we can treat the hemophilia by isolating the gene, by introducing the gene responsible for the clotting factor 9 to be introduced into the patient target cell. So by using retroviral system, the gene for the synthesis of factor 9 were inserted to the liver cell of dogs. These dogs no longer displayed symptoms of hemophilia. So first time it was not done in patient, it was not done in human beings. Human trial is not done, it was done in dogs. What we have see that the dogs which are suffering from hemophilia, what we have done, we have used the clotting factor 9 gene to be introduced into the liver cells of the dogs via retroviral vector system. So we have used retrovirus as a vector, and we have inserted our desirable gene into that vector. Obviously we have used some selectable marker genes for observing that whether it is getting to be expressed or not, to check out which of the cells are getting transformed, and we have introduced them into the liver cells of the dogs, and we cultured them in the laboratory, and then we will again reintroduce them into the dog's body, target cells, target tissues. It was seen that the dogs were no longer suffering from hemophilia. So that's quite interesting. and now it is trying trial is going human trial is going to be done by gene therapy to treat hemophilia patients. Okay, now what can I say about here it is saying the gene therapy with non-autologous cells. So ex vivo gene therapy with non-autologous cells. Non-autologous cells means cells from other cells. Okay, by using other species cells actually. So it is saying here that ex vivo gene therapies described above are based on the transplantation of a genetically modified cells for the production of desirable proteins. So in the previous examples, like for curing SCID, for curing hypercholesterolemia, for curing the hemophilia, for curing the sickle cell anemia, uh, ex vivo gene therapy was done, and it was based on transplantation of genetically modified cells for the production of your desirable proteins. But there are some limitations in using the patient's own cells. So if you are using the patient's own cell, there are some limitation for the gene therapy. And what are that limitation? Lack of enough cell from the target tissue, defective uptake of genes, and their inadequate expression. So the problem arises that from the patient we are obtaining the cells, na, and we are using that cells to be transfected by our gene of interest via vector. But some limitation is there, like lack of enough cells. We don't get enough cells from the target tissues. Defective uptake of the genes, that means most of the cell it is found that it is not able to uptake that genes, the normal genes. And inadequate expression, they are not able to express that genes properly. Now, to overcome these problems, we are using non-autologous cells. That means we are taking cells from outside source, like from other individuals or from other animals. Like I have said, that human artificial chromosome, right? They can be used as vector. How it can be used as vector? We have to take from other individual cell, and from that cell we have to take that chromosome out. We prepare a human artificial chromosome, introduce our desirable gene, and then it is targeted to the patient. So from non-autologous cells, that means cells taken from other individuals or cells taken from animals, we can introduce our desirable gene to be expressed, and then it is to be incorporated into the target tissues of the patient. Okay, so in this way we can go for the gene therapy by using non-autologous cells. Now, how it can be done, how the cells from other individuals or animals can be used in the gene for ex vivo gene therapy, an outline has been given here with the in these two paragraphs, in this one paragraph. So let us try to explain. Tissue specific cells capable of growing in culture are selected. So the tissue specific cell which can be grown in the culture medium within the laboratory are selected, like your fibroblast cells from the skin, like hepatocytes from the liver, like myoblast from the muscle, and astrocytes from the brain. So these are the four cell sites where we can use them to culture. Like we can take fibroblast cells from the skin to be used to culture within the laboratory. We can use the hepatocyte cells from the liver. We can use myoblast cells from the muscle and astrocytes from the brain. So these fibroblast, hepatocytes, myoblast, astrocytes can be used uh, as a non autologous cells. Okay, they can be used as a non autologous cells to be used for the gene therapy. Then these cells, after isolating these cells, they are cultured and genetically modified with our therapeutic gene. So by isolating these cells, any one cells out of them, if you are targeting to cure rectify certain disease related with liver, then we have to isolate the hepatocytes. If we are trying to rectify a disease which are related with muscles, we have to take out the myoblast from the other individual, or astrocytes from the brain. Okay, and after that, what we do after isolation of these cells from the other individuals or animals, then we are just we have seen in ex vivo gene therapy where we are culturing that cells and genetically modified the cells with our therapeutic gene. These are then encapsulated in artificial membrane composed of synthetic polymer. Now, after these cells are cultured with a therapeutic gene, now these cells are encapsulated, means entrapped within an artificial membrane, and that membrane consists of a polymeric substance, synthetic polymer substance like polyether sulfone, alginase poly, L-lysine alginate. This type of synthetic artificial membrane polymer is being used to entrap that cells by this artificial polymeric substance. So if this is your cells, this is your artificial polymeric substance. Okay. Now these polymeric substance are not immunogenic. Okay, they will not causes any immune response to the patient after delivering. Therefore, patient can accept non-autologous encapsulated cells. Further, being semi-permeable in nature, this thi- this synthetic substance, this polymeric substance, the synthetic polymer that we have used to encapsulate the cells are semi-permeable, okay, and thus this membrane will allow the nutrients to enter in and encoded proteins by therapeutic gene to be passed out. So what we have seen here, let us see in short brief. It is saying that we are taking from the other individual or from the other animal, we are taking out the certain tissues, certain cells here we are taking out, which can be used to culture them in the laboratory. Like we can isolate the fibroblast from the skin. We can isolate the hepatocyte from the liver, myoblast from the muscle, and astrocyte from the brain. Okay. After isolation of these cells, what we can do? We can culture them within the laboratory. We modified them with therapeutic gene. Okay. By using vector mediated approach, either by using human artificial chromosome or by using retrovirus, right? Then what we do? Then they are encapsulated, then these cells are encapsulated. That means if this is your cell, and if this is your target gene which has been inserted, they are then encapsulated, means coated with a synthetic polymer. Now this synthetic polymer is semi permeable. That means what? It can allow entry of the nutrients and an exit of the products into the cells, and this synthetic polymer is not immunogenic. It will not create any immune response, okay, to the patients after delivering them, okay, and thus it will make mediate the functions normally, easily, okay. So experiments conducted in animals has been shown this type of benefits. Some encouraging results has been observed using these non-autologous cells in gene therapy. Encapsulated cells were found to proliferate and produce the required protein. However, success has been very limited in human trials. So in this way, by using the non-autologous cells, that means by using the non-patient cells, we are using some individual cells, we are isolate, we are using and culturing the cells from taken from animals. We culturing them in the laboratory, and then we will entrap them, we coated them with synthetic polymer so that no immune reaction will occur when these cells are going to be introduced to the patient. No immunogenic response will occur.
And these synthetic polymer are semi permeable also. They are semi permeable, they can allow the entry and exit of the products from the cell. Okay, so in this way, from this approach we can use a one step more improve in the ex vivo gene therapy. But animal models have been prepared, that's good, and it is quite success. But in human trials it has not met with much success. It has been, the success has been limited, okay, in human trials. So this was all about your ex vivo gene therapy. I hope you have understood this part. If you have any doubts regarding this ex vivo gene therapy, let me notify me, ask me through the WhatsApp group. Okay, study this gene therapy part within today, and for doubts, you have to comment in the WhatsApp group so that I can able to understand that what problem you are facing, and then I will try to explain them either by WhatsApp audio or by through text. Okay.